Once upon a time, there was a little mermaid named Ariel. She met her prince, Eric, and they had a daughter whom they named Melody. One day, Melody was wondering why her hair color looked like her dad's. Eric then explained to Melody the process of protein synthesis. Before Melody's gene for her hair color can come out of the nucleus, it has to be transcribed. Transcription is the process of making an RNA copy from a DNA template. There are 3 steps to transcription: initiation, elongation, and termination. During initiation, RNA polymerase binds to a location on DNA called the promoter, which signals the start of transcription. RNA polymerase has the ability to unzip the DNA just like the helicase in the process of DNA replication. The next step of transcription is elongation, during which RNA polymerase makes a complementary RNA strand from one of the exposed DNA strands. This DNA strand is called the antisense strand, and as the RNA is copied, it begins to have a sequence of bases that code for amino acids. A set of 3 mRNA bases in a row is called a codon, and it codes for a particular amino acid. Because there are 64 codons but 20 amino acids, one amino acid can have multiple codons. The final step to transcription is termination. During this step, RNA polymerase reaches a DNA sequence called the terminator that stops the transcription process. Free mRNA has now been made before it leaves the nucleus, however, it must be processed. In the editing process, introns, which are sequences that don't code for anything, are cut out. The exons, which do code for a protein, are spliced together. Finally, a cap and a poly A tail are added to the mRNA. It is now ready to exit the nucleus. The next step to determining Melody's hair color is translation. Translation is the process in which a ribosome reads the mRNA and makes a protein or polypeptide. The initiation of the translation begins when the small subunit of the ribosome attaches to the mRNA and travels to the initiation site. Then tRNA and its anticodon bind to the start codon, AUG, on the mRNA. The tRNA also contains the corresponding amino acid to the codon, in this case methionine. Finally, the large subunit of the ribosome binds to the mRNA to create the P and A sites. As translation continues in the process of elongation, the next tRNA binds to the mRNA in the A site of the ribosome. Its anticodon matches with the next codon on the mRNA. Methionine attaches to the amino acid on the second tRNA, and the first tRNA floats away. The second tRNA then moves into the P site, and the third tRNA moves into the A site. The amino acids bind, and the second tRNA floats away. This process, sometimes called the assembly line, continues. Finally, during termination, the ribosome reaches a stop codon, either UGA, UAG, or UAA, and a release factor enters the A site. Translation is terminated. The ribosome floats away, and the newly formed protein, which determines a trait, in this case hair color, is released. Melody will have dark hair. However, if a mutation had occurred, Melody's hair color could have been red, like Ariel's. There are 3 kinds of gene mutations that could cause this. Gene mutations are also called point mutations because they affect a particular gene only. The 3 types of point mutations are substitution, insertion, and deletion. In Melody's case, let's say that a substitution occurred, making her hair red instead of black. If the adenine base in the codon sequence was replaced with a cytosine base, it would cause a change in the shape of the protein and thus a change in the function. So instead of making Melody's hair black, it could be red, like Ariel's. In the end, after Eric's explanation, Melody finally understood the great mystery behind her hair and how protein synthesis affected its color.